Thanks, Rafa.
Thank you for the introduction, and already thank you for the nice discussions. It's always nice to see people from different fields, jumping at this. So let me see if you see my presentation and everything looks reasonably okay. Yeah.
Yep.
Excellent.
Good.
So again, I'm going to talk about remote access to equipment, in particularly from the perspective of a microscopy core facility. As it was mentioned, I'm coming from the Center for Cellular Imaging. So we are a national core facility that integrates both light and electron microscopy. And we are basically a part of the national microscopy infrastructure. So in that sense, following a little bit the discussions, we actually offer open access to our equipment. And that means researchers from universities or companies in Sweden, but also from abroad, right? And in this sense, we are also part of the Euro-BioImaging Swedish node. Again, we have a plethora of different equipment and different services. They can go in from the light microscopy all the way from wide field. So that's basically a microscope that works in a similar way to what your eyes would look like. So they have cameras and so on. And then we go into confocals and high content screening, super resolution, and so on and so forth. That's still visible light. But then we also have electron microscopy. And then, of course, this works in a slightly different way. The samples look completely different. And at the same time, we have now a project on multi-imaging mass spectrometry. So now what you are trying to do is that you are trying to also image kind of chemical compositions of the samples. And one thread that connects all of this is image analysis, of course. Now what you see at the bottom is all my colleagues, and you will see that we have a quite broad spectrum of kind of backgrounds. And this is actually the strength at the facilities, that we have multimodal, not only equipment, but also staff, and then that we love to take the same sample and move it between different equipment. And we like to basically try to get as a holistic view as possible from the sample, right? That aspect that we are an open access, and I like to term it in a way like user-centric facility, that basically means that the way we work with our users is that we try to train them in the operation of the machines of the microscope in such a way that they become independent as soon as possible. After that, they can have access to the microscopes 24/7. Of course, this will depend on the level of training and clearance, right? So depending on how experienced you are, you start to get sort of upper level of access to different types of equipment, and also in different types of working hours. Not only we offer training to the equipment, but we of course offer continuous support via project consultation and training and so on and so forth. So we are constantly monitoring the projects to make sure that everything is running as expected, or if the users have any problems, they can contact us. Now, since the pandemic, we actually changed the way we are training our users. So we used to have completely hands-on, physical trainings, but now we actually have divided our training into 2 sessions. This is for everybody, not only for the remote users. Is that we have an initial remote session of 2 to 3 hours, depending on the equipment, where we are actually going to focus on the software. And we have seen that this is actually incredibly useful for everybody, because we are recording these sessions and we make these videos available afterwards to the users. So in most cases, people find these videos very, very valuable, because then they can go back and double check later on if they have missed something or they don't want to write it down. After this remote session, then we have, for typical users, we have an on-site session where we focus on the hardware, and then also on specific things about the user sample. We tend to do all our interactions that are remote via Microsoft Teams. This has to do due to our IT infrastructure. And the microscopes are actually within an intranet. They do not have any access to the internet. We control everything via proxies. So we actually can have a lot of control over who can have access to the microscopes and to which type of networks the microscopes are exposed. And then basically takes us to this point of remote services. So now we have sort of remote activities that are part of our everyday work, but we also have specific remote services that we can use with our users. One of the most common, let's say, is this remote support to on-site users. So the idea is that in this case, the operator, which is the user that is sitting at the microscope, might encounter some kind of problem, is able to communicate from the microscope computer to the staff and ask for help. Sometimes this help can be provided on site, but sometimes we can also just log in remotely, have a chat with the user, and try to help them that way. The second way that we are doing quite a lot is this acquisition guided by user feedback, right? So sometimes if we have a researcher, as you might know, like geographically, Sweden is a very long country, right? So people might be very far away from us, but they want to work with us. What we try to do is that when possible, that they will ship samples to us. We put it in the microscope. We test the samples beforehand to make sure that we can actually provide service for them. And in this case, of course, there is nobody like the user to know the biology, right? So we need their input while we are looking at the sample, so we can actually have a good idea of if the project is going to be successful or not. And then the last level is this complete remote instrument control. And the idea here is now the operator is going to be a remote user. And this we're going to demo a little bit later on during the workshop. Now, there is other type of remote services that I will just hover over, which are related to data management and image analysis support. But those are perhaps more kind of classical in a way because we're interacting via servers. So this is perhaps much more, straightforward or common in the field. Now, one of the things I want to mention is the fact that all of these things are possible just, I would say, due to the fact that we have established a very clear data management plan for the facility. So we have a very good idea of where I hope you can see my cursor because I'm sort of pointing at things. So we have very good control over the different networks of the facility. So the acquisition and CCR is within a specific network. They only have access to the things we want them to have access, and then afterwards they will transfer that data that is acquired at the microscope via different transfer servers to the different user data solutions. So we already had all of these sort of workflows in place, which allow us to adapt very quickly when we wanted to give remote access, because already the users were not using standard ways of, no USB drives, no data drives, no hard transport of data between one place to the other. We also had a lot of experience doing image analysis in a collaborative fashion, so this also helped us a lot to change our services. All right, and if you have any questions later on about this, I can explain a bit more. Now, remote instrument control. So this is the part where everybody in a facility gets stressed, especially in a microscopy facility, is that setting up a truly unsupervised remote instrument control is very challenging, and you get really, really scared. What you see here on the right is called a microscopist's nightmare. So it's a series on Twitter, which I'm very fan of, that they show in how things can go wrong in many, many, many different ways. So what you really, really want to avoid is that situation, right? So there is some general considerations that you have to, keep in mind, which I think is also not only for microscopists, but for many different fields. And one of them would be the safety of the equipment, right? So we have to make sure that the equipment is going to be saved and that the user will not be able to damage it by an unfortunate accident, right? Now, if we can guarantee that the equipment is going to be safe, then the next thing that we have to fix is our IT infrastructure, right? And this really, really depends on the host institution. We have been having discussions with different colleagues around, and each institution has a different way of handling this. And then there is a few things in there to keep in mind, such as, okay, how are we going to make sure that the communication speed between the different parts that are involved are correct? And also, how are we going to then manage the data so it can be efficiently transferred between different store solutions, different image analysis servers, and finally to the user? So if we have these two things in place, then we can start also thinking about a training procedure, right? So how are we going to train users who have never been here at the facility potentially to use an equipment that they have never touched, right? So we have to find ways, efficient ways to train our users. And then I think one of the biggest questions is remote control really needed? And what we have found is that in many cases, actually just remote monitoring of experiments might be enough. So as it was discussed already, the more automated the pipeline that you are trying to expose to users remotely, the better in a way. So if you have enough automation, usually what the users want to know is to be able to log into the machine, see what is happening, and perhaps make decisions such as this experiment is working, is not working, should we change samples, not change samples, things like that. And that basically also will allow things such as democratization of resources, again, to researchers that are not close to us. And I already briefly mentioned this, which is like, if we manage to have automation built into these pipelines, then the easier it is to give remote access to them. In terms of the democratization of resources, we are also trying to set up ways in which we can allow for external users to use our machines via shipping of biological samples, which is not always straightforward, but in some conditions this is possible. Also, we have situations where we have live cells running, but the researcher, for example, might live in a city a little bit far away, and they might just come during during very specific days and times, but they don't have to be moving around with the cells all the time. We actually argue that preparation of biological samples at the source and shipping them to the CCI is often more efficient, and actually it allows to re-implement different models that we have at the facility. And actually, I think that it will allow for a better reproducibility of the data, because you have very specific tasks that are taken in one place that have to be very well documented. Then it goes to the next stage where you have to do the imaging and so on, and that actually helps quite a bit. Just glossing over this idea of smart microscopy is just to say that there has been a lot of effort recently to connect image analysis, so like how we can identify features on images and so on, with fully motorized and computer-controlled microscopes. And when I'm talking about smart microscopy, is actually the idea that these two things can talk to each other, so that the microscope can look at the sample, have an idea of what is happening, and then with that, they will make decisions onto how to proceed with the experiment. Now, smart microscopy as a service is quite challenging. Now we believe that that there has been a lot of solutions provided by the community, but sometimes these solutions are very specific to a particular use case or a particular research group. And then we believe that advanced microscopy core facilities such as the CCI can actually take these concepts and make them available to a research community as a broad community. And then we're very proud that recently we got funding for this project for the next 5 years. And on that note, we're also recruiting for this. So if you are interested in this sort of automation and remote access, you can always reach out to us. With that, I wanted to cut it a little bit short, just to have a lot of time for discussion. And I, of course, want to say thank you to everybody, especially to the team, to the Center of Cellular Imaging, here my colleagues, and then the people that have given me funding. And with that, I'm very happy to take any questions that you might have. So thank you very much.
Thank you for the presentation,